about out-of-the-box cloud solution for next-generation sequencing analysis.
Yes, my name is Freerk van Dijk. I'm a PhD student in the group of Maurice Sweats at the University Medical Center Groningen, and I will tell something about our virtual machine image we've developed for NGS analysis. And yeah, we came to this project because we have several sequencers in our lab, and we also participate in large projects like Genome of the Netherlands, where we had 770 individuals sequence whole genome, and we ran into the huge issue that we had well, a large pile of data, and we wanted to do reproducible analysis on it, like alignment, variant calling, SV calling, etc. And we also quickly ran into the scalability issues, like Piotr also already mentioned. So we started on a local cluster processing all this data, soon scaled up to the national grid, and eventually thought, well, why don't we use the cloud? And by doing this, making a virtual machine image would also allow biologists to easily do their own analysis. So that's what we focused on with this project. So as I already mentioned, we developed a virtual machine image, and in four small steps, user can start their own NGS analysis. And basically it's these 4 steps. So the user downloads the image, starts it, installs software and resources, uploads a sample sheet and the data to the VM, and can generate jobs which are then run inside the VM. So for this we used, for the first step, we used the OpenStack framework, and we basically choose for this because it has a easy dashboard which is viewed inside the browser, and you can basically just start your VM, assign some resources to it like CPUs, RAM, etc. And it has a wide community behind it, this active development. And once you've started the VM, you only have to run 2 shell scripts which automatically fetch all the software and resources. And to actually ensure additional reproducibility, we decided to use the EasyBuild framework, which is a Python package developed by the university in Ghent. And as the authors state, you need to build recipes, and these recipes basically describe which compiler you need to use, which version of dependencies, does some sanity checking, etc. Everything is included in this VM. The next step for the user would be to upload their data into the VM, and we wanted to keep the metadata and stuff lightweight, so we decided to let them specify this in a CSV file. And basically this CSV file, you could enter sample IDs, project names, the sequence it was run on, flow cells it comes from, etc. And that already brings the user to the last step, which is generating jobs. And what I didn't mention yet is that all our pipelines are available in GitHub. So for now only the DNA resequencing pipeline is in the VM, but we also have pipelines available for RNA-seq, imputation, genome builds, liftover, etc. Everything according to best practices. And you can generate your jobs using the Multihonest compute framework. And this actually is a framework we've developed in-house. And to generate jobs, you basically need 4 things as an input. So one is the sample sheet, which I just showed. Another sheet of parameters. Then a directory containing templates, and these templates are just plain bash scripts containing all the commands you'd like to execute. So in this case it could be a command to do alignment, run GATK, BQSR, whatever. And the user needs to specify a workflow file, which also is a CSV file. And this workflow file just contains the names of the scripts you'd like to execute and the dependencies between them. And with a push of the button, the software then generates these scripts. And these scripts can be run inside the VM using the Slurm scheduler or just sequentially. And within the framework we also have support for BSUB, PBS and other schedulers. So yeah, then the conclusion, we've created a reproducible, a VM image which makes the research reproducible. By using OpenStack, it's quite scalable, and it also makes it portable and easy to use for biologists. And everything is available over here in the bottom. That's it.